Catholic University, Nursery Career, Valencia Aro Kerli. Study of ventricular Cerliponase Alfa for CLN2 disease. Definition: Neural cerlipofunctionosis type 2 disease, a form of Bayern disease, is a rare autosomal recessive pediatric neurodegenerative disease resulting from pathogenic variants in the gene encoding lysosomal enzyme tripeptidase peptidase.
This is a cell in Mila's brain, and this is a lysosome, which breaks down the cell's waste. But in Mila's cells, the lysosomes aren't working, and they start to fill up. The cell tries making additional lysosomes, but it eventually becomes overwhelmed and dies. It turns out the culprit is a mutation buried deep within Mila's DNA that affects the lysosome proteins. To make a protein, the cell uses its DNA as a blueprint to make RNA by splicing together the essential bits of code. The cell machinery then reads the RNA to build the protein. But in Mila's RNA, a misplaced piece of genetic code has thrown off the splicing process. So when the cell's machinery comes to assemble the protein, it's halted by the misplaced code and the produced protein is defective. Thus, Mila's lysosomes can't function properly. Researchers at Boston Children's Hospital have designed a special drug just for Mila. It's called Milacin. The drug hides the mutation in Mila's RNA with a new piece of genetic code. This allows the RNA to be made correctly, which lets the cell produce normal proteins. The lysosomes can now take out the trash, and Mila's cells stay healthy.
Application in nursing. Cerliponase Alfa is administered by intraventricular infusion at a rate of 2.5 mm per hour. An antihistamine drug is administered approximately 30 minutes before its infusion, a phase of increasing the dose in which the drug starts with 30 mg, 100 or 300 mg.
What we try with CLN2 patients is to see them with a multidisciplinary team. It's not only the pediatrician, child neurologist, epileptologist. We also ask the physical therapist to come and see these patients. We talk to the caregivers. We spend a lot of time talking to the parents because we want to know what their daily life looks like, how they are doing, in order to find out whether there are any medications that have side effects that they do not tolerate well. Overall, we try to not give them too many different medications at the same time. We try to limit the number and also the dosage of the medication. The goal should be to really have the best quality of life that we could get in this patient. And quality of life also means to be awake during the day, to participate in daily life and not to be sedated most of the time. Sometimes you cannot prevent it, but usually our experience is that if we are careful with medication, we have a better effect. And also, we try to be critical with our own decisions. It might be that a couple of months ago we have started a medication and we see the child again and we have to decide that this is not the correct medication for this individual. We have to do something else. Or this medication was the right one for a number of times, but at this time there seem to be the side effects that are not tolerable, so we should change that. So it's not always the disease that shows symptoms or side effects or things that are a burden of disease, but it's sometimes even the medication that we provide. So we should always be critical and also be courageous enough to stop medications or change them.
Pros. Cerliponase alpha, a form of recombinant proenzyme, is an enzyme replacement therapy that has potential use in patients with CLN2 disease. The administration of enzymes in the ventricular cerebrospinal fluid were spontaneous. Homologues for TPP1 deficient resulting in weak spread, distribution, and uptake in the brain. Cons. The natural history of children with CLN2 disease have shown a progressive decrease in motor function and language. A database of children with the disease as characterize the severity and progression of the disease, which include motor function, language, and visuals, and incorporate the frequencies of epileptic seizures.
This is TPP1, short for tripeptidylpeptidase 1. This little enzyme has big responsibilities in the body, and the lack of it can lead to big consequences. TPP1 is a lysosomal enzyme that helps break down proteins in the lysosomal lumen. Specifically, it cleaves the N-terminal tripeptides from a broad range of substrates. When functioning correctly, the proenzyme form of TPP1 is synthesized in the endoplasmic reticulum and makes its way through the secretory pathway to the Golgi complex. Here, the protein gets an important modification. Oligosaccharide groups, such as mannoses, get added to the protein during protein modification. An enzyme called N-acetylglucosamine phosphotransferase in the Golgi recognizes these proteins and transfers a phosphorylated N-acetylglucosamine group to carbon-6 of one or more mannose residues. Next, a phosphodiesterase removes the N-acetylglucosamine group, leaving the phosphate group behind on the mannose. This is considered a mannose 6-phosphate targeting sequence that helps deliver enzymes to the lysosome. M6P receptors in the Golgi membrane are responsible for recruiting enzymes with these targeting sequences and forming a vesicle to transport them to the endosome. At a higher pH in the Golgi, the receptor has a higher affinity for the M6P sequences, but at a lower pH in the late endosome, the receptor loses its affinity for the targeting sequence and the enzyme is released into the lumen. Additionally, the lower pH is responsible for activating the TPP1 proenzyme, also known as a zymogen, to its active proteolytic form. Once the late endosome becomes the lysosome, the TPP1 is able to degrade proteins using its catalytic triad active site to hydrolyze peptide bonds. 3 amino acids are brought together by protein folding and carry out ping-pong catalysis, involving binding a substrate, kicking off the N-terminal, binding water, and releasing the C-terminal end. However, if a mutation occurs in the gene that codes for TPP1, the enzyme may not be synthesized, it may not get delivered to the lysosome, or it may fold incorrectly, inhibiting its catalytic action. In patients with a TPP1 deficiency, proteins like ATP synthase subunits cannot get degraded once transported to the lysosome and begin to build up in the cell. When proteins accumulate in nerve cells, it can lead to progressive decline in motor function, vision, and language, as well as cause seizures. This is a genetic disease known as CLN2 disease that mainly affects children as they develop and can lead to premature death. CLN2 disease symptoms can be slowed with a drug recently approved by the FDA, known as Brineura, or generically as Cerliponase Alfa. Cerliponase Alfa is a replacement enzyme for TPP1 that is administered to the patient in the form of TPP1's proenzyme. It is able to be taken into the cell via receptor-mediated endocytosis using M6P receptors that naturally reside on the cell's surface to reuptake any lysosomal enzymes that accidentally get sent out of the cell. Like the natural form of TPP1, Cerliponase alpha has M16 targeting sequences that allow it to bind the receptor at higher pH and dissociate from it at a lower pH as it is transferred to the lysosome. Here, the enzyme is activated and it is able to cleave peptides to prevent further buildup. After a little while, the replacement enzyme is degraded and the patient must be administered another dose of Brineura. While it does require continuous treatment and it is not a cure, Brineura has helped many families see a slow progression in their children's.
Issues apply in Ecuador. Affected children are functioning normal until the age of 2 to 4 years and secondly have seizures and delayed language acquisition, followed by a rapid decrease in motor language, cognitive and visual function over a period of 4 to 6 years and at the beginning of adolescence.
I felt the necessity to have a system to describe what we call the clinical course of the disease over time in terms that can be compared from case to case, from patient to patient, from hospital to hospital in a quantitative way. So we wanted to develop some scoring system that really works. You have to be able to go back, you see a patient, and then to go back, how did his or her disease develop? And we tried to develop questions to be asked to the family, simple questions that the family can answer in a reliable way. We tried many different questions, but it turned out certain things are remembered very exactly remember very well at what point in time the child lost the ability to walk independently. This is a sharp moment in time, not only in the individual child, but also in the souvenir, in the memory of the families. We developed such scoring points that the parents could develop and tested several questions, and it turned out what we now call the clinical Hamburg scores. It is a really primitive looking thing of simple questions, but questions that can be remembered very well and can also, as we say, prospectively used to follow the disease and describe it with numbers 3, 2, 1, 0 for every few months. And so over time you get the picture how the disease evolves.
Conclusion: intraventricular administration of cerliponase alpha every 2 weeks at a dose of 300 mg in children with CML2 disease results in a slower rate of decrease in motor function and language than in historic controls. The potential use of ventricular cerliponase alpha to prevent the onset of symptoms in young patients.
Cerliponase alpha. It was approved for a lysosomal storage disease called as infantile neuronal ceroid lipofuscinosis. So this condition is basically due to deficiency of an enzyme called as TPP, tripeptidyl peptidase. So basically this is a recombinant TPP enzyme. So with deficiency of enzyme is there, so we give enzyme from outside, so deficiency will be treated. Now first thing we are discussing here TPP. Where did you read DPP, dipeptidyl peptidase? Yes, DPP4 in particular. Yes, this is the enzyme which cause metabolism of GLP. So if we inhibit this by drugs like gliptins, we can use in diabetes. So TPP is involved in lysosomes. So deficiency of this lead to infantile neuronal ceroid lipofuscinosis, and cerliponase alpha is a replacement therapy. And again from the name you can remember ceroid, CER is ceroid, lipo, ceroid lipofuscinosis, neuronal. Okay. So neuronal ceroid lipofuscinosis, we can use cerliponase alpha. It is given by intracerebroventricular injection. It is not given orally or IV. It is directly injected inside the ventricles of the brain. Okay.